So here we have the calcium IP3 and DAG pathway right here. And so it starts here with the signaling molecule binding to the G protein receptor. And the G protein is then going to work on the enzyme in the complex. And the enzyme here is the phospholipase C. And what phospholipase C does is it takes a phospholipid from the membrane, a specific kind called PIP2, and it cleaves it, and it's going to turn it into or convert it into two different molecules. One of those molecules is called DAG. There are some bigger words that these are abbreviations for, but we only need to know the abbreviations. So one of these molecules that the PIP is converted into is DAG. The other is IP3. IP3 is the molecule that we are following. IP3 is then is the second messenger, and it is going to move to the membrane of the endoplasmic reticulum, and it's going to open up the gated ion channel or the gated calcium channel, and which is going to release calcium. Calcium is a second messenger. You could call it a third messenger, but we just call it a second messenger. It's just another second messenger. So this second messenger is then used to elicit a response or to activate or deactivate another protein, which will elicit a cellular response. So that is part 2 of our notes about signal transduction pathways. Let's go through to our next set of notes, which we have 1, 2, 3, 4, 5, 6 slides. So this will go, or 5 slides, so this will go super quick. And so here we're at the last part of our pathway, which is the cellular response. And a cellular response can be many different things. So we talked about epinephrine. So it could be to release epinephrine, or it could be to release, what epinephrine does is it breaks down the glycogen into glucose. That could be the cellular response. It could also turn on or off a gene by starting to start transcription. So there are 4 aspects of cellular responses that we're going to investigate. One is amplification. We talked a little bit about that in the last set of notes. Specificity and coordination with other signaling pathways. Efficiency by presence of scaffolding proteins. And termination of the signal. So how does it end? So let's start looking at amplification. So here, multi-step pathways amplify. We said that it's really important that these multi-step pathways are that we have these signaling proteins and we have multi-step pathways. So here we have a signaling protein that is going to activate our G protein, and the G protein is then going to activate. It can activate up to it says here 100 adenylyl cyclase molecules, and then the each adenylyl cyclase can activate many cyclic AMP, and so each cyclic AMP can activate many kinases. So this is going to make the message move quicker and more efficiently through the cell. This amplification can happen because the proteins stay activated long enough to work on that amount of substrate. All right, next, specificity and coordination. So these multi-step pathways allow for specific and coordinated steps. So epinephrine is going to affect different types of cells with different responses. So for example, a heart cell, when it receives epinephrine, is going to lead to a more rapid heartbeat. Liver cells, though, when it receives epinephrine, is going to break down glycogen. So these are specific responses. And when we are talking about coordination, the signaling molecules can have different effects even within the cell, and this is going to allow for multiple things to happen at once to coordinate maybe different responses or responses that you want to have happen at the same time. So here we have our first example, cell A, and we have our signaling molecule and our relay protein and our response. So this is our basic outline here. And so cell 2 is going to show us a way that coordination can happen. We can have our relay molecule, and what it can do is it can act on different enzymes or different relay proteins, which are going to elicit multiple steps or responses. Next, we have cell C. This is called crosstalk. It occurs here. You can have multiple signaling molecules which will elicit the same response. So this relay protein will be able to activate this enzyme just like this protein can. And so the same response will be elicited by both of these different signaling molecules. Finally, in cell D, we have a different, notice that this protein here is different. It's a different shape, and that indicates that it's a different type of receptor protein. And so while we have the same kind of signal being sent, signaling molecule, it's going to be received by a different protein, which is going to send a different set of instructions, or it's going to create a different pathway and elicit a different response. So that is these are the ways that specificity and coordination occur in our cellular responses. Efficiency occurs by scaffolding proteins, and this is pretty cool. We have these proteins here. This big red protein is a scaffolding protein, and it's going to hold multiple other proteins that are in the transduction pathway, so that a message can be sent 1, 2, 3, boom, 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 more quickly. And so this scaffolding protein holds everything in place, but it can also activate and deactivate these proteins as well. So this helps to make the responses more efficient. And finally, termination. What's really important about our multi-step transduction pathways is that a message can only last a short time. The molecule needs to be able to be turned on and off, or the signal needs to, the signal needs to be able to be turned on and off. So we saw the effects of cholera occurred when the protein, or when the message was not able to be turned off. So there was salt and water continuously being released into the lumen of the small intestines, and in that scenario, homeostasis was lost. So we need to be able to activate and deactivate these transduction pathways so that the signal can stop when it's stopped, but also change when it needs to change. So it'll be turned on and then turned off again, but if the signal remains still present, it'll turn back on again, but then turn off, so that it's ready to either end or to change or to continue on. And so that makes it a more effective way to have a response. All right, so that is the end of chapter 11. I will see you guys in class.